Okay, hi, this is Laura Niklason from Yale University. I'm sorry I can't be there today.
Hello.
I guess I should begin. I'm assuming that the first slide of my presentation is up and showing. I guess I'd like I guess I'd like to ask for the next slide. So sort of related to the previous talk that you just heard, my group has been working for the past 4 or 5 years at Yale on developing techniques to engineer functional lung tissue. Consistent with the spirit of the meeting, I'd like to provide a little bit of background here on why anyone would want to do this. In the US, lung disease is the fourth leading cause of death. But unlike the liver and unlike many other organs, the lung actually has very, very limited capacity to regenerate beyond the microscopic or cellular level. So once the lung organ is damaged at the tissue level, at the level of a segment or a lobe, that tissue never regenerates in the human, and probably not even in young humans, in children. So after severe lung disease is incurred, either through injury or infection or smoking, really the only treatment is lung transplantation. But lung transplantation suffers from really high morbidities, with only about a 20% survival at 10 years, and which is much worse than for many other organs. And the primary reason for this is either slow rejection of the transplanted organ, despite immunosuppression, or the occurrence of severe and repeated infections in the transplanted lungs. So in view of all of that, if we had the ability to regenerate lungs that contain cellular elements that were not foreign to the recipient, essentially growing autologous lungs, then we're anticipating that these transplantation survival rates would be much better. Next slide. So as a tissue engineer or bioengineer, it's always good to think about sort of the key design criteria that you want to build into anything new that you're trying to make. And so in that spirit, I would say that generally there are about 4 key criteria for engineering a lung successfully. First is that it must have the right mechanical properties. In other words, it can't be too stiff, so that it's hard for the patient to inhale and exhale, but it can't be too floppy and compliant, because that type of lung is actually an emphysematous lung, and that doesn't function very well either. The cells in the construct should ideally be autologous, so that we avoid the whole rejection issue. There must be adequate surface area for gas exchange, so that the lung can subserve its most important function, which is exchanging oxygen and carbon dioxide. And there also must be adequate barrier function in the lung, so that the air components and the blood components are separated from each other, so that when you implant the engineered lung, you don't get flooding of the airway compartment with elements from the bloodstream, because that obviously doesn't help you with gas exchange. Next slide. So just to do a little bit of lung physiology, it's really interesting to think about lung structure. If we think about the normal human lung, we start off with the trachea that then branches into 2 bronchi, and then those bronchi subsequently branch, and then those branch again. And in fact, there are 23 generations of branching in the human airway. And after all that branching, the airways terminate in literally hundreds of millions of air sacs, which help for gas exchange. And each of these air sacs is about 200 microns in diameter. And the total surface area of all of these air sacs is on the order of 70 square meters, which is just a tremendous surface area. And so as we began thinking about trying to regenerate lung a number of years ago, we considered for a while trying to develop a synthetic scaffold that would allow us to culture cells and regenerate lung. But as we thought about it, as we thought about the anatomic complexity and the huge surface area and the requirement for the correct mechanics, it just became pretty clear that we would not be able to generate a synthetic polymer scaffold that would have all of these characteristics. Next slide. And so we developed an alternative paradigm, which is similar to what you heard in the last talk, which is where we utilize a donor lung scaffold, which has been decellularized. So this is an adult lung. Most of our work so far has been in rats. In the work I'll describe, we decellularize a native adult rat lung, and then we repopulate it with pulmonary cells. And for our rat work, these cells are syngeneic. In other words, they're taken from different members of the same species into which the implant will go. And in fact, not just same species, but these rats are closely related enough that they're almost genetically identical. Although in the future, as we envision this for human work, these cells would be autologous, and I can talk more about that towards the end. But this decellularized scaffold then, which is shown in B, is mounted in a bioreactor, and the bioreactor is fitted with several apparatuses that allow us both to breathe the lung during culture using negative pressure ventilation. So we're essentially mimicking native breathing, particularly native breathing in the fetus, which continuously breathes in amniotic fluid during lung development. We also have a perfusion system that allows us to perfuse culture medium through the decellularized scaffold so that we can provide nutrients to the lung. And then we have means of seeding cells, both vascular endothelial cells into the vascular compartment of the lung, and then epithelial cells into the airway compartment of the lung. So after mounting the decellularized scaffold in the bioreactor and maintaining this culture for a period of roughly 7 to 10 days, the organ is then removed, and it looks roughly like what you see in D, and then that organ can either be analyzed in vitro, or in some cases can be implanted in vivo into a syngeneic recipient. Next slide. So to give you a sense of what these scaffolds look like, you'll see in the upper left there's an H&E slide of a native rat lung, and then on the right a decellularized rat lung, showing that the nuclei obviously are absent. When we quantify DNA content, obviously most of the DNA is removed in the decellularized scaffolds. Next slide. Because the structure of the gas exchange surface and the integrity of the vasculature is so important for this application, we've actually developed a number of new techniques that allow us to directly query how intact our acellular scaffold still is. So on the bottom left there, you see a black and white image, which is actually a scanning electron micrograph of a decellularized rat lung, where we've injected a contrast agent into the airway and then digested away the rest of the lung, so that we're able to take an SEM image of the air sacs that are remaining. And as you can see, after decellularization, the air sacs are all still pretty intact. Alternatively, we can examine the vasculature, in this case by micro CT scanning, again by injecting a contrast medium into the vasculature and letting this harden, and then doing a CT scan. And both of these assays show us that at least at this level, sort of at the micron level, or tens of microns level, that these structures appear to be intact. If we do a little bit more assessment of the acellular matrix, we were very interested in understanding the biochemical composition of the matrix and whether or not we were retaining the key matrix molecules that we think are important for lung mechanics and function. And so one of those molecules is elastin. As many of you probably know, elastin is very important for spontaneous recoil of the lungs. When elastin gets degraded, actually that's an important component of lung emphysema, and so we want to avoid that, obviously. And this is one area where we're probably still not good enough. So if you look at the stains for elastin in the native versus the acellular lung, the elastin appears as sort of black wavy lines or dark wavy lines, and it appears on staining as if some of our elastin is degraded. When we quantify this by biochemical assay, we've seen that we lose about 60% of the elastin in our matrices, which is something that, frankly, we're continuing to work on. That being said, the collagen seems to be retained, but the glycosaminoglycans and, as I've said, the DNA are all largely removed. Next slide. So I mentioned as one of the key design criteria the fact that we needed to maintain adequate barrier function between the air sacs and the microvasculature. And what's fascinating about the lung microanatomy is that surrounding each alveolus, each air sac, is literally a net of capillaries which surrounds the air sac and which allows very close approximation between blood and air so that we can get highly efficient gas exchange. And these are some scanning electron micrographs of vasculature. So the two top images show casts of the vasculature, and the image on the upper right actually is an image taken looking into an air sac. So that network that you're seeing in the image in the upper right is literally a network of capillaries surrounding the air sac that obviously looks fairly intact, and the scale bar in that image is 20 microns. So each of those microvessels is on the order of about 8 microns in diameter. However, although those images look pretty good, there are clearly some areas of the lung where we, not intentionally, but we disrupt that barrier function. So if you look at the image on the bottom, you can see a vascular cast, but you can see areas where the injected vascular contrast has leaked into the air sacs. So again, this tells us that our decellularization processes, while pretty good, are not perfect. And so in these areas, those would be areas that would be at risk for getting flooded with blood components after transplantation. Next slide. So I'm going to move away now from just sort of describing and characterizing the acellular matrix, and I'm going to move and describe some of the steps that we go through to repopulate that matrix to try to regenerate something that's like a living lung tissue. So the photo that you see on the left is a bioreactor with culture medium in it. Those large black things that you see are actually just one-way valves, but the engineered lung is deflated and it's sort of in the middle of the image, and you can see cannulas going into the pulmonary artery and into the trachea. And on the right are pressure tracings of pressures that we record from the perfusion on the upper right, and then from breathing on the lower right, which shows that the pressures that we are delivering are actually very similar to native pressures, both for breathing and perfusion. Next slide. So this slide just provides a little sort of simple diagram of some of the different subsets of cells that are in the airway. Airway epithelium comes in multiple flavors, and airway epithelium changes from the proximal airway in the trachea all the way down to the alveolus. So in the proximal airway, there are columnar epithelial cells, which have cilia, and there's a local stem cell population in green, which is the basal cells, and those cells renew the columnar epithelium locally. Going further down the airway, there are more cuboidal cells called Clara cells, and these cells are only moderately well understood, but there's probably a subset of those cells, also shown in green, that are a local stem cell to renew Clara cells. And Clara cells make secretory proteins and also help with airway clearance. In the bottom of the airway, in each alveolus, there's two cell types. One cell type is the type 1 alveolar epithelial cell, which is a very thin cell and which lines the alveolus and helps with barrier function, helps keep water out of the alveolus. And then there's another local stem cell type called the type 2 cell, which is a precursor cell for type 1 cell, but the type 2 cells also make surfactant. And surfactant is very important for lung inflation. It's one of the things that makes it easy for us to breathe. So I'm going to be talking about each of these cell types in subsequent slides. Next slide. So as I've said, for our experiments on rats thus far, we've isolated epithelial cells from neonatal rat lung, and we've characterized these cells and then implanted them in the matrix. So this is just FACS data showing populations of different types of cells that we obtained from neonatal rat lung, including Clara cells, cells that make surfactant protein C, which are type 2 cells, cells that make aquaporin 5, which are type 1 cells, as well as some other cell types. It's important to note, though, that when we seed these epithelial cell populations into the rat lung, we don't do any cell separation steps first. In fact, what we're doing is we're seeding this entire mixed collection of cells directly into the airway. And the results that we've seen have actually been fairly interesting from that. So if we just, next slide please. If we just look at the histology of reseeded lungs when the experiments are working on a good day, if you look at native lung and then repopulated lung, they actually look pretty similar. We can get pretty broad distribution of cells throughout the airway, and their viability is quite high. The biggest difference between these two images is the image on the left. You can clearly see red blood cells in the microvasculature in the native lung, but in the repopulated lung, obviously there's no red blood cells because we have no blood in the bioreactor. Next slide. So if we look at the, again, sort of looking at a higher level, if we look at the compliance or the mechanics of these engineered lungs as compared to native, one good way to do that is to do pressure volume loops, where we take a lung and we inject air into it and we measure the pressure that it takes to inflate the lung. And if you look at the native as compared to the decellularized lung, what's interesting is that near these arrowheads on the upslope part of the curve, you can see that for the decellularized lung, that slope is much shallower. And what that means is that it requires a lot more pressure to inflate that lung at low volumes. And that increased pressure requirement actually stems from the fact that when we decellularize the lung, we also wash away the surfactant. And the surfactant is what makes lungs easy to inflate. So what's interesting about the engineered lung is that one thing that's interesting is that after a week of culture, the engineered lung regains some of that ease of inflation, meaning that functionally, physiologically, some of the cells in that lung are making surfactant, and we're getting some of the compliance back. Next slide. So if we look at immunostaining for different cell types, all of these data are captured in lungs that have been cultured for 4 to 7 days and have received mixed populations of cells into the airway. And what we're going to do is we're going to look at immunostains for cells going from the bottom of the lung up toward the top. In most of these cases, the immunomarker is red and the nuclei are blue. If we look at this stain for aquaporin 5, which is a type 1 epithelial marker, what you can see is that in the native lung, the red marker really outlines the alveoli, showing that the cells are really spread out and really outline the whole air sac. The green dots in the native image are actually autofluorescing red blood cells. But if we look at the reseeded image, in fact, you can see that in at least some of the air sacs, we also see this very spread out morphology of these type 1 cells. And we never see this type 1 cell marker in larger airways. We only see this in the distal air sacs. Next slide.
Laura, you have 2 minutes.
If we look at surfactant protein C, which is a marker for type 2 epithelium, also in the alveolus, you can see that in native lung, we see these rounded oval-shaped cells that sort of sit in the corners of the alveoli, and this distribution is actually remarkably similar in the reseeded lung. And when I initially saw some of these images a number of years ago now, I was really taken aback because seeing these images really told me that this lung matrix that we're seeding must have a lot of information in it. It must have zip codes or local protein information, which actually tells cells where to land or tell cells who to become once they get there. And we're frankly not sure which one of those it is, but we're working on experiments to sort that out. But it was really this image that first made us think that there are zip codes, that there are local instructions in the matrix that dictate where different cells will land. Next slide.
Laura? Yeah.
Okay, well then I will try to finish up really quickly. So this is clear cell secretory protein, which is going further up the airway. You can see that repopulated lung is very similar to native. Next slide. This is actually looking at the impact of perfusion and ventilation on the engineered segments. We don't have to talk about that too much. This just shows that when we ventilate these lungs, the secretions in the airways are actually washed out, meaning that the airways that we're seeing are contiguous with the trachea. Next slide. This is staining for alpha-actin positive cells, which are mesenchymal cells in the lung matrix. This just shows that they're present in native lung, but also in the engineered lung. Next slide. And then this is staining for cytokeratin 14, which is a marker of basal cells, which are the local stem cells for the proximal airway. We don't see many columnar ciliated cells in our engineered lung, probably because we don't breathe very much with air, and that's probably required for differentiation. But we do see some basal cells, shown here in red. Next slide. This is just an image of the microvascular endothelium. So this is a stain for CD31, which shows that we also get dense repopulation into the vasculature of endothelial cells. And these are cells that we directly inject into the vasculature, and they take up residence there and form a very dense population. Next slide. We can skip over this. This is some quantification of barrier function, just showing that the barrier function in the engineered lung by some of our assays is only moderately good, and it's clear that we still need to improve barrier, both by improving the matrix, but also improving our endothelial and type 1 epithelial cell seeding. So I still think we've got a ways to go there in terms of how dense those cells are. Next slide. So I'm just going to finish up now with the lung implantations. We've done a number of implants into rats. This is our microsurgeon, Tai Yi, who's a very clever guy. And what he's done is left lung transplantations of our engineered lungs into syngeneic recipients. Next slide. This is just a photo of the engineered lung at inflation and deflation. And as you can see, it at least grossly, it's similar in appearance to the native right lung. Next slide. And you can see a chest X-ray of the implanted lung, showing that it's aerated and well inflated. If we look at the saturation of oxygen that's emerging from the left implant pulmonary vein as compared to the right pulmonary vein, in both those cases the blood is completely saturated. The PO2 in the right pulmonary vein is somewhat higher than in the left, but because most of the oxygen is carried on hemoglobin, this difference in PO2 actually corresponds to only about a 5% difference in oxygen content. So what this tells us, what all this data tells us essentially, is that the implanted left lung was actually very efficient in gas exchange, almost 95% as efficient as native lung. And so that was the good news. But everything has a downside, and the downside here was that after a few hours, what happened was the vasculature in the engineered lung began to clot off. And we think that's at least partially because our endothelial lining in the vasculature wasn't as good as it should be. So after a few hours, we started to get thrombosis, which limited our gas exchange, obviously. And when we took the lungs out, we also saw some evidence of red blood cells in the airways, as you can see in the lower right, meaning that our barrier function was probably not as good as it should have been. So in conclusion, it appears that the acellular lung matrix does provide remarkably specific cues for cell repopulation, but the matrix is still somewhat leaky, and we need to improve the uniformity of the repopulation. But even at its current state, the repopulated lung is implantable and reliably exchanges gas. Every single engineered lung that we've implanted has exchanged gas efficiently. We've never sort of had a failure there. But maintaining blood flow through the lung and improving endothelial cell coverage are still things that we obviously have to improve. Then I'd just like to acknowledge my outstanding group of folks who worked on this. Tom Petersen and Elizabeth Calle were really the people who've driven this project. They've been helped by Lee Chong-Gui and Alice Lee and Li Ping and Sumati Sundaram. And I apologize for going over. I'll take any questions.
We have about 1 minute for questions, so you've got a really important question.
Hello, if I understand very well, to make the decellularized organ, you use a chemical approach. Why don't you use an approach like propoptotic drug in a very high concentration? Maybe this could avoid the disruption of the matrix.
I'm sorry, you use what at a high concentration?
Sorry, why, if I understand very well, to create an organ without cells, you use a chemical approach, not a pharmacological approach. Have you ever think about the possibility to use a pro-apoptotic drug to destroy the cells in a biochemical way and so leave the scaffold intact? Is it possible?
Well, what worries me there is that there would be elaboration of proteases, which might break down the matrix. So part of the decellularization process actually relies on speed, removing the cells and their proteases quickly before the matrix gets damaged is actually important.
Okay, thank you.
Well, sorry for no more questions. I don't want Aubrey to get really upset with me for not keeping this meeting on time.